Okay, let's get started. I hope you attended the short course last Friday, and Michael mentioning about the background, NGS, next generation sequencing, and the resources available on HPLC for data analysis. So this short course will be a specific topic. We'll be talking about how to do genotyping with sequencing data on HPLC. So my name is Shichen Wang. I'm a bioinformatics scientist in Genomics and Bioinformatics Service Center. So here's our website. In case you're interested, we are in AgriLife Research, and our website is textgene.tama.edu, and we provide genomic sequencing, whole genome, all kinds of sequencing we have. So we have Illumina, the latest Illumina sequencers like Novo 6000, we got 2 of them. And we also provide a bioinformatics service. So we're going to talk about next generation sequencing, NGS. So mainly when we're talking about NGS, we are saying DNA sequencing. Because even if you are doing RNA-seq, the RNA will be transformed into DNA to do sequencing. So the early method for reading the DNA, you probably know the Sanger sequencing and all those things. And that was called the first generation of sequencing. So that's why when we have this high throughput sequencing available now, we call them next generation sequencing. So the several companies are providing this NGS service, like sequencing by synthesis method developed by Illumina. And that is the most popular sequence method available on the market now. Another company called Ion Torrent Sequencing, I think they are part of Thermo Fisher now. They have a different way to do sequencing, and we generate a different kind of data. And there are small companies like Genopsys. They're trying to break into this market by developing a little bit lower throughput, but much cheaper sequencing solutions. Okay, so that is generally the part of next generation sequencing. It has been a very long history, how the method was developed, different companies, some companies come and go, some companies just out of business. So the survivors like Illumina, our Torrent, and they are the competitors on the market now. So if you're thinking doing next generation sequencing, basically there are only like 2 options for you. Either you go Illumina or you go Ion Torrent. So like Genopsys, the small company is trying to develop a new machine. So they're still working on the machine, so it's not released yet. And you probably heard about like PacBio sequencing, like a long-read sequencing. So that is the long-read sequencing technology. And another long-read sequencing is like a nanopore sequencing, Oxford nanopore. You heard those Million and those really long, give you really long reads. So those are part of the long-read sequencing technologies, and probably some people call it the 3rd generation sequencing. So it's probably that, but today we're going to be focusing on the NGS, the new and the next generation sequencing. So let's before we start going into details, let's see how in general the NGS sequencing the NGS works. So you start from the sample collection. You get the samples, then you extract DNA or RNA from the sample. You do quality control. Okay, so next step after quality control, there'll be library construction. So you have to use the kit provided by the company. So it depends on the sequencer you'll be using. So you're going to be using different kits. Also depends on the how you want the sample to be sequenced, how long the read you want. It's DNA or RNA or small RNA. So you have all kinds of kits available for you to choose. Okay, so after library construction, basically we can say just load the library into a sequencer. So the sequencer can probably run like from 4 hours, 24 hours, or even longer. Then it will give you sequencing data. So what you get, just the ATGC sequences in short fragments. So after you got data, then you need to run the bioinformatics analysis to get some insight, to get some biological meanings from those data. So okay, so this is very general. So if you're looking at the sequencing types, you can do whole genome sequencing. If you want to focus on the exome, it's fine. You have the exome targeted sequencing. Even more than that, if you want to only focus on a few genes, a few regions, we have the amplicon sequencing. So let's say I want to do a whole genome wide, I want to do a genome wide sequencing, but I don't want to do a whole genome sequencing. I don't want to capture whole genome, but I want the region I want to be sequenced are like more or less evenly distributed along the chromosomes. So you can do a reduced repetition library type of sequencing. Some probably heard of the RADSeq or GBS. So if you are interested in DNA modification, like methylation, those kind of things, yes, bisulfite sequencing is also available. So the popular ones like RNA-seq, you want to measure the gene expression levels, or you want to assemble the transcriptome, you need to run RNA-seq. So there are many other applications available yet. Yeah, you just need to explore. So there are a few concepts are kind of important in the next generation sequencing. The first one is the read length. So the sequence, the sequencer will give us the data in very short fragments. So those sequences, we call them reads. So one read might be 75 base pair. If you're doing sequencing back to 5 years ago, it's probably a 75 base pair. Now the read is much longer. It can go to 100, 150, 250, or even 350. I mean, for Illumina platforms. So obviously, the longer reads can provide more information for you, but it's really more expensive. So you need to think about your applications. Do you want more read or longer read? So it's like a compromise. You have to think about that. So another concept in this sequencing is single end or pair end. So in single end, the sequence read, the fragment is only read by machine once from one end. So the pair end read obviously means it's a sequencer going to read a fragment from both ends. So let's say if you have a fragment that is 250 base pair, and you are sequencing single end 150, so the sequencer will give you 150 base pair from the left end and read 150 base pair. That's what you get. But if you do pair end, the sequencer is going to read 150 base pair from the other end. So you got a 150 plus 150 is 300 base pair longer than the 250 base pair of the original fragment. So you're gonna have a 50 base pair. You're gonna have an overlap there, so you can actually connect those two reads and make them single read much longer than the 150. So pair end is important because it can give you, obviously, first is more information, more sequencing there. Second is you can use a pair end to, let's say, if I have a fragment that's really long, that's like say it's a 6,000 base pair, I do 150 on one end and 150 base pair on the other end. When I map those two reads back to reference genome, I can see, because I know the fragment might be like longer, might be 6,000 base pair. So if I map those two reads back to reference genome, I expect to see them far away. The distance is supposed to be like 6,000 base pair. So it's one way to help you align the read correctly to reference genome when you have pair end reads. So if the distance is not 6,000, let's say it's 200, whatever. So that means something happened in the reference genome, right? It could be a fragment being deleted in the reference genome. So the many informations can be extracted from pair end reads. That's why it's really popular. Okay, so the third concept is called coverage depth. The coverage depth is like how many times that region being sequenced. If I have a 100 base pair region, and I have 10 reads cover the whole region, so the coverage depth will be 10x, because each of the bases in the fragment will cover 10 times. So why we need a higher coverage depth? Because sequencer produce errors, some random errors. So some cases you're gonna call some bases wrong. So you have an A, it's actually a G. But if you have 10 reads cover the same location, you will clearly see that, because the A won't show up 10 times. It will probably show up 1 times A and 9 times G. So to give you some clue, maybe at A is an arrow. And okay, so a very brief introduction how the libraries are made, because I'm not working in the lab, so this is, I feel like this is enough for me as a bioinformatician to know how the data was generated. So basically you have the DNA insert. It is the fragment you want to be sequenced. So to make this fragment sequenceable, in the library, during the library preparation step, you need to add those adapters. So P5 adapters and P7 adapter to the fragments. So on the adapter, once the adapter is added, you have this finished sequenceable fragment. It has the primer, you have the index, sometimes it has the adapters, all those things. So this is a sequenceable libraries. Then how it works on the Illumina machine, there's a flow cell has a pre-attached oligos that are complementary to the index on the libraries. Let's say the red fragment here, that could be an oligo that's attached to the flow cell surface. So once it's attached, and the sequencing called sequencing by synthesis. So you need the primers start synthesis, and you're gonna release fluorescent signals, and each signal represents different allele, different nucleotides. So, and the machine gonna detect a signal and tell you is it A or T or B, G or C. So if you're doing pair end, so this one end sequencing when it's done, you're gonna flip over and attach the oligo to another pre-linked oligo there, and start sequencing the second part of the fragment. So this is a very general introduction how this library works, and I feel like for a bioinformatician, it's already good enough for me. So we say the technology for Illumina is called synthesis by sequencing by synthesis. So every step is incorporate the nucleotide in the synthesis step. You're gonna release it's a light signal, so different colors. So this is old chemistry, back to 2 or 3 years ago, and this is old chemistry. So every nucleotide got a different color. So you see A, T, G, C, different colors. But now with a new chemistry for the Novo Assembly 6000, and Illumina only use 2 colors. So 2 colors to encode 4 nucleotides. So it's possible, right? 0, 1, 0, 0, 0, 1, 1, 0, and 1, 1. So it's 2 colors. It's already good enough. Okay, so let's say you know all those background, but you don't really need to. The only thing you need to pay attention is when we got the sequencing data back, how it looks like before you start doing analysis. So most of the time, you're gonna be looking like a list on the top of the figure. So you see on the left hand is an adapter. It's an adapter, and then some bluish color here. It might be a barcode. Then you have your DNA that you're interested in. You want those to be sequenced, and it's probably possible another barcode at the other end, another adapter at the other end. So when you got the data, the adapters mostly will be gone, because the machine, when the machine do a demultiplexing, it will remove the adapters. And if you provide a barcode for each sample, and the machine gonna do the same thing, it's gonna multiplexing. Basically, it's gonna tell you, you're gonna split a lot of reads into different samples. Sample 1 based on the barcode, and you're gonna tell those reads from sample 1, those reads from sample 2. So you're gonna receive a lot of files named based on the sample's name. Okay, so the data will be stored in a format called FASTQ. So basically it's a text format, and each read was represented by 4 lines. So I have an example here. The line 1 we have start with the @ sign, and it has a machine name and the flow cell name and the tire number, all those things you don't really need to pay attention to. And you have the index nucleotide there. So, and have a /1. /1 means it's a single end. It's a read 1. If we are doing pair end for another file, you're gonna see /2. That means it's read 2 because you are doing pair end. Okay, so the first line is just the information line. It got the machine name and all those things. And second, and so the first line is the name, so it must be unique. And the second one is the sequence. You see it's ATGC. Sometimes it's have bad qualities or the machine couldn't determine what nucleotide is, so it's gonna be ends. So the third line, it is the same as the first line, but except the first character is not the @ sign, it's a plus sign. But other than that, it's all the same. So because of that, many times you will see the third line just a plus sign. There's nothing else. So the fourth line is the quality. So for remember, machine has some arrows. So the Y machine gonna tell you how confident machine think this place is this certain nucleotide. And the confidence is represented by a score. And so instead of using quality scores like 1, 2, 3, 4, a nucleotide numeric thing there, it uses characters. So have E, F, C, F, those things. So there's a way to convert the characters back to scores. There's a formula to do that. And many tools can do that, so you don't need to worry about that. So here is an example here. I have this is a screenshot from my login to the ADA machine. So I check this file, vj something fastq.gz, and only check the the 10 lines. It's only show 4 lines here. So you see the first line start with the @ and then following all those informations. And the second line is the sequence itself. The third line is just a plus sign. And the fourth line is a score. Okay. So how do we convert the score back to how can we convert the character back to a score? So there are different scales. You see the Sanger scale is from, if you look at this plot, it's from 0 to 40. And it's going to be using those symbols and numbers and A, B, C, D, F, G, H, I. So if the FASTQ file is using Sanger scale, you're going to see the best quality will be represented with a letter I, the capital I. So the most popular one now, most likely, if you are doing sequencing, most likely you will get the Illumina 33 scale. So it's going to be the last one. So from 0 to 41. So it's very close to Sanger. It's just one more letters there. Okay, so we know. So let's see the 31. The 31 is the @ sign. So and have the capital A, B, C, D, F, G, H, I. So let's go back to the previous slide here. So see the A, B, C, D, F, G, H, I. We know those are scores. I think more than 30, right? Probably 33, 34, or something like that. The F, G, H, I is the highest score. So the I is 41. Okay, so that's how you convert to the score. But what does the score means? The score, the quality score there is an integer mapping of the quality of the probability P. The probability P means the probability that the base cast colony is wrong. So let's say I have a Sanger score Q30. What is the P? So based on formula, you can calculate the P will be 0.001, 0.001. So it's like 1 out of 1,000% chance this base is wrong. So it's a very small chance. So normally we say if we see the quality score 30, it's a very good score. Okay, so before we can, now let's say we see FASTQ data, but before we can start to analyze the data, we need to know a few more file formats used very popular in bioinformatics. The first one is a FASTA format, just one letter then different than the FASTQ. So it contains a sequence name, a description of the sequence sometimes, and the sequence itself. So whenever you see the greater than sign, and following by some letters, those will be sequence names. So in this case, I have a greater than then chromosome 1. So chromosome 1 will be the sequence name, and all the information after chromosome 1, chromosome down to 4, those things will be the description of this, those sequences. And after that, we're gonna multiple lines of sequence, a real sequence, ATGC, all those things. So if you have a reference genome, the reference genome will be in this FASTA format in most of the cases. So another thing is, another file format important is the SAM or BAM or CRAM format. What are those for? Those are used for to show the alignment. So we have the read, let's say I want to map the read to reference genome to figure out the differences between my sample and the reference genome, right? So how to represent those alignment? People invent this format called SAM or BAM, and later one the CRAM. So in the SAM or BAM format, you have the data was separated into columns. So column 1 will be the name, the sequence name. Second, column 2 will be flag and tell you how this align went. So it's unique or it's reverse complementary. All those information will be there. And the third one will be reference name. So this really goes to chromosome 5. So you will see chromosome 5 in the third column. So a lot of information is there. And when we have a chance, we're gonna take a closer look at the SAM file itself. So one more thing is the GTF or GFF3. So it's still text format, and it's a tab delimited text format. It has information about the genes, the structures, the gene structures, the CD, the coding sequence, the exons, all those things. So those are all gene annotation. So those will be saved in the GTF or GFF3 format. So here example here like this. Let's say if I have those gene structure like this, the orange part will be the exon, and the gray part of those part will be like UTR or something. So those will be represented in the GTF file, and you're gonna see first column will be say in this case will be CTG123. That means either it's a contact name or the chromosome name, the first column. You have a dot sign, then have the gene. Gene is a feature name. So we know, okay, this is gonna be the gene, and it has a location start from 1000 and in 9000. And it has an ID equals something, ID equal gene name, and it has a name. So those are the informations for this gene itself. And more details for this gene, if you keep looking, gonna give you about the mRNA, the exon, the CDS, UTRs. So you're gonna see those informations in the GTF and the GFF file. So that file is important because when you have the variation discovered, you want to know if this variation is located within a gene or outside of gene. So within a gene will be more interesting because it's cause a function change, might cause a function change, right? So one more thing, it's a variation calling format, a VCA format. It's also a text file. So when I say it's a text file, it means it can be opened with Notepad or UltraEdit or those simple text editors. So in bioinformatics, most of those files actually just text file. So this one, the VCF, is the same. It's a text file, but it's a tab delimited. And it is used to describe variations. So like let's include single nucleotide polymorphism like SMP or the insertion short deletions. So let's take an example here. Okay. So in the header of this file, usually you see a lot of lines start with the hashtag. Those are just for annotation, except the last hashtag here you have here. So those lines with 2 hashtags are just the annotation as a comments. So, and then there's one line with 1 hashtag. That is the line has the important information saying chromosome, position, ID, reference allele, alternative allele. Okay, and quality filter information and format. Then following by the NA, those are sample names. So see, that's why it's important. It has the sample names there. Okay, so after that, you're gonna see the real variation information. Let's say, let's take a look here. Have a 20, that means chromosome 20, position 14370, that means it's at least position. The ID can be arbitrary, you can assign anything to it. Okay, now 2 important things. The reference, the REF means the reference allele, and the ALT means alternative allele. So reference allele is allele in the reference genome. Alternative allele it's a new from the samples from the sequences you provide for alignment. So many cases, you have one allele for the reference and another allele for the alternative. But let's look at this number, this line here. You have the reference allele is A, alternative allele have G and T. So that means in your samples for that location, you see two alleles, G and T. So sometimes this could be wrong because either it's because of sequencing error or either because of wrong alignment. You have the homologs somewhere else has the T allele, and because the alignment couldn't find that location, or in the reference genome, that part is gone. So the T allele from the homologs were aligned here, and that's why you see A and G and T. So yeah, those are information you can pay attention to when you have to analyze the result, analyze the variation calling results. Many cases we will just ignore those multi-allele variations because first we don't know if it's true or not. Second, it's most likely many cases is because of the wrong alignment or sequencing errors. Okay, so a summary here. We talked about the FASTQ format. We talked about the FASTA format. So if you have a reference genome, it's gonna be in FASTA format. And for the gene annotation, they'll be stored in the GFF or GTF format. For sequencing data you're gonna get from the sequencing facility, it'll be in FASTQ format. But many of cases to save storage, to save space, those will be zipped. So most of the cases you will see a FASTQ.gz. And alignment result will be saved in the SAM format or BAM format. So SAM and BAM are the same. So SAM is a text format. BAM is a binary SAM format. So it's like a compressed to save space. So CRAM is a new format, and it's contain more information than SAM and the BAM, as it's taking a smaller space too. But it's not very popular right now. The variation will be saved in VSAVE format. Okay, so if you are doing a variation data analysis, those are the format you'll be dealing with. So what do we need for a high throughput variation discovery analysis? So first, we will need to have a reference genome. That would be a perfect ideal situation, because then we can map the reads to reference genome and just compare the difference among samples, right? But what if there's no reference genome available? If that's the case, I would say you're not really lucky, but there are ways to do it. First one is a Novo Assembly. It means you do a whole genome sequencing at 30x, at 550x or 60x coverage depths, and trying to assemble it, the genome. Genome assembly is kind of a difficult job. In many cases, you won't have a full chromosome assembled. You'll get a lot of pieces, a lot of fragments. So another approach is called RADSeq or restriction associated DNA sequencing. So this was a very popular one, and I think it's still popular. So the idea is, instead of sequencing the whole genome, because I don't have that reference genome, so instead of sequencing the whole genome, I try to pick up enzymes and digest with a specific enzyme, and only sequence the fragment that's close to this enzyme cutting sites. That's why it's called restriction associated DNA sequencing. A different protocol developed for this, and people call it genotyping by sequencing. So just different names. The idea is mostly the same. So this way, you're not sequencing the whole chromosome or whole genome. You only sequence a small percentage of the genome. But you see, because the distribution is supposed to see a random distribution of the distribution site, right? So we can say, yes, it's not whole genome sequencing, but we are sampling genome-wide variations. So it still can be called genome-wide variations. If you're doing a RADSeq GBS, when you're doing, let's say if you want to map this read to reference genome, they're gonna look like this, very unique, very even coverage for each locus. But if you don't have a reference genome, you can still do it by just clustering those reads, because those reads, they are from the same location, same cutting site. So those read from this site, based on the sequencing sequence similarity, you can cluster them together. Then you can call variations within the cluster. So another really popular genotyping sequencing approach is called scheme-seq. It's the same idea as the RADSeq, but because some pattern issues or something, people are moving away from RADSeq to the scheme-seq thing. So this one is really rely on a good reference genome. So you're gonna do a whole genome sequencing. It cannot be the RAD sequencing. It'll be a whole genome sequencing, but you're gonna be really shallow. That means you'll not be sequencing at 50x or even 30x. You'll be sequencing at maximum 5x, or sometimes you can go lower, like 1x. So then the analysis can be the same for most of the part, except when you have the variation code, there's gonna be a lot of missing data there, a lot of missing in the final genotyping matrix. So one way to improve that is using imputation methods. You try to impute the missing data in the genotyping. So that's why this scheme-seq can work. If you have a reference genome. Second, if all the samples in the population, you got to have a lot of samples for this kind of project, have 300 or 500 samples and sequence together. So if the samples are in the population, they are like closely related. Let's say I'm doing a breeding. I use two parents and cross them and produce a lot of progenies. I'm sequencing those progeny with low depths. That's fine, because those progenies gonna help each other to fill in those missing data. This gonna be a successful imputation for this. So we're gonna fill in missing data, then we can use those data for the applications. So you see, the important part here is the imputation, and it really depends on reference genome. Is a good reference genome or not? Second is the population itself. So this is the detail how you can analyze this data. I think we're gonna run on the hands-on practice session. Gonna do this the whole pipeline. So let's get a little more details about pipeline. So first step, when you get the data from the sequencing facility, you want to do first thing you want to do is quality control. So we want to first check the qualities with a tool called FastQC. I think most of the time the facility is going to provide you a FastQC report. You can just check if they're good or bad. If there any problems that you can deal with, that you have to deal with in the quality control step. So what we normally do at this step is we're going to remove the low quality bases. Remember, in the FastQ we have a quality score. So we can say we're going to pick up a score, say a 20. So 20 will be 0.01. There's like 1 out of 100 chance to be wrong. So if it's less than 20, we say it's low quality. You're going to trim them. So 20 is arbitrary. Sometimes people pick 10 or something like that, just to save more data. And in many cases, if you look at the FastQC report, you're going to notice if there are any adapter sequence left in the read. Why there are adapter sequence left in the read? Because some fragment might be very short. Let's say a fragment itself is only 100 base pair, but you are doing 150 sequencing. So after the 100 base pair, the 50 will be the adapters because the fragment itself is too short. So we need to pay attention to those, and we want to remove them. So second, after those trimming and adapter removing, some reads are going to be really short, like less than 30 or something. So those will be removed too. I mean, because first, when it's too really short, it's hard to run alignment to get the correct alignment for those. Second is, if you are doing a de novo assembly, those short reads will not help at all. And the pair end information, we're going to keep the pair end information correct in the correct way. So the tools can be used like FastX, Toolkit, Trim-O-Matic, a lot of tools. So those are all open source and free to use. So I think we're gonna use Trim-O-Matic in our practice. So, and Trim-O-Matic is a Java program. So it's how you start Java program here. It's Java -jar and geometric .jar. And you're gonna give the mode, it's pair end mode or single end mode, the quality score scale and the input and how you want the output to be. So here you have the leading 3, it means you're gonna cut the first 3 bases away, because if you look at, if you have been looking at the FastQC report, you will notice the first couple of bases usually have low quality. It's somehow related to the chemistry. And trailing, you can remove 3 couple bases from the tail. And sliding window approach. Okay, so here, this is the quality control step. So we're gonna use the 4 bases sliding window and using 15 as a quality cutoff. So let's say I have a first 4 and I calculate the average quality for those 4 bases. If it's less than 4, cut those 4. Less than 15, those 4 bases will be gone, will be trimmed. Then move to the next 4. If it's already higher than 15, then I stop. The trimming stop there. And the minimum length, say here is 36. So those numbers are, it's not like those are golden standard, and you can change, say, starting window to 5 and 15 to 10. I mean, it won't change very much. The latest sequencing technologies has very good accuracy. So I won't worry too much about quality for the recent technologies. Okay. So after the QC step, now you're ready to map the reads to reference genome. Let's assume you have reference genome, you're really lucky. So this step is trying to figure out where in the genome that those sequences are from. So this is a very tricky stuff. It's the most time-consuming step in the whole analysis pipeline. So first, the reference genome can be really big, right? If it's a human genome, it's 3 GB, and it's gonna take a long time to search. If it's a plant genome like wheat, it's gonna be even bigger. It's like 16 or 17 GB. So it's gonna take a lot of time to search for small things. And you will be looking for, the program will be looking for exact match. No, we will not be looking for exact match because there will be variations. Sometimes have SMP, sometimes have indels, right? So that makes it even harder. So good things, we already have many tools being developed, like a BWA, very popular, and Bowtie2 and something that are very popular. So I have this plot in here. This is the tools developed just for doing alignment. You see how many different tools are being developed. This is the most active bioinformatics research part. Okay, so for the alignment, there's 2 concepts we just need to understand. And when we go to the practicing session, you're gonna see we need to pick one. So one is a global. So global alignment means you're trying to match the sequence to reference in to make the full sequence aligned. But local, you're just trying to find a subsequence of high similarity. Let's see here. So if you have a really long read, that means the local alignment will be meaningful to you, right? If you have a short read, you can do a global alignment. So, okay, so we will be using Bowtie2 for the practicing session. So the reason is because Bowtie2, I have been testing Bowtie2 and BWA. So in terms of performance, like accuracy of things, they are quite similar. And but Bowtie2 is a little bit faster and memory efficient. And it has been pretty good at aligning reads about 5 to even longer, like 1,000 base pairs. And before we use Bowtie2, we need to make index of a genome. So you're gonna produce, you have to process the genome first. And Bowtie2 support gapped alignment, local alignment, and pair end alignment, and the global alignment. So gapped means you can discover the indels. Okay, so if I type in Bowtie2, let's say if I log into the machine and type in Bowtie2, it'll give me a lot of information about how to use this program. So you need to provide the index for the reference genome. You need to provide read 1, read 2. If you are doing pair end reads, if you are doing the single end read, just use -u, and all those things, different ways, and output. So we're gonna see more details. Okay, here's the thing when we need to pick the global or local alignment for both two. And they have some predefined parameter set for end-to-end. So end-to-end is like a global alignment. And for local, it's like a local alignment. So for short read, you can pick end-to-end, is fine. For longer read, you can pick local. Okay, so once we have the alignment, let's say we have the reads and we feed those reads to BODAI2, BODAI2 give what they're gonna give us. You're gonna give us a SAM file. It's an alignment file we talked about before. And what we need to do, we can use that file to call variations. So we need to, several steps we need to process for that before we can run variation calling. So first we need to, if it's SAM, we need to convert to BAM, to binary SAM. Then we need to run sort the BAM file. So the reason is you're gonna sort based on the coordinates. So chromosome 1 will be the first you see, and chromosome 1 position 0 if position 1, if there's any read aligned there, you're gonna see them from the first part of the file. So you're gonna sort the BAM file based on the coordinates. Then you need to add the read group names to BAM if necessary. So why? Because sometimes you have a lot of samples, and each sample is gonna have has its own BAM file. So if you don't put a read group name to the BAM file, and at the end step, at the end, you're gonna put all the file together and call in variations, and the program will not be able to tell which file is for which sample. So we need to add the read group names to a BAM file. Basically, it's like a sample name to the BAM file. Mark duplicates. Okay, so this step depends on how your libraries were made. If you are doing a whole genome sequencing, it's totally random. You're not supposed to see a lot of PCR duplicates. So those duplicates will be removed. But if you are doing like a RADSeq or GBS, you know a lot of reads gonna be from the same restriction cutting locus. So they are duplicated. So you don't do this mark duplicate step. So local realignment. So what this is something like when you have map a read to reference genome and it's detect a gap. But because of the gap, the boundary of gap is really hard to define. So that's why we added one more step here called local realignment. That means we take the sequence that have the read that has gap when we compare to reference genome and run a closed local realignment to figure out how those gap boundaries are defined. So there are a lot of false SNPs actually are caused by this wrong gap boundaries. Okay, so after that, one step you can do is filtering with mapping qualities. So mapping quality, the program, when you like a BODAI2 or BWA, it's just trying to tell you the best possible part this read is from, right? So how to define the best possibility? Basically, it depends on how many mismatches you got from this alignment. If I put this read here, I got 10 mismatches. If I put this read here, a different location, I got 0 mismatches. So obviously, I should put this read to that location that has 0 mismatches, right? So it's more likely it's from the 0 mismatches location. Okay, so based on the mismatches and based on how many locations you can put this read to, let's say I have this read, I can put this read to 10 locations that are perfect match, that has no mismatches at all. So that means I have 10 locations that's equally best. So, so the uniqueness here is not unique, right? Uniqueness here is very low. It's like a 0. So the uniqueness of the mapping will be considered into this mapping quality too. So if this read has a lot of location can be mapped, the quality will be low. And if the mapping has a lot of mismatches, the quality will be low. So we can use a mapping quality to remove those reads that are poorly mapped. So we don't know like a golden standard for this mapping quality. Sometimes people use 30 for SMP, 30 for the mapping quality, or even 20 or 20, 30, 40. I've seen many people using different numbers. So many of the cases you can do is probably just take several alignment and visualize it on the tools like IGV and check if it's the 30, what you got with the 30 and the 40 and the 20, get the general ideas. So and they just pick up number. I mean, there's no absolute number you can use. Okay, so next thing is we can visualize using tools like IGV to see if the alignment are good or bad. And in many of the cases, our eyes actually can grab the arrows quickly. So if you see some arrows in the alignment, what do you do? Like then you need to go back and adjust some parameters here. So you want to try, maybe you were using fast or very fast, but the accuracy might be low. So or you want to choose to sensitivity, very sensitive, 2 different parameter sets and give it another try. Okay, so after all those steps, finally we can call variations, right? Variation, many of the cases when you are using a short reads, you'll be focusing on the single nucleotide polymorphisms, or called SNPs, or small indels. Here, example here, let's say you have 2 samples, one at this particular location, one has A and one has G. So this might be a single nucleotide polymorphisms. So the purpose of this step is to figure out all the locations that has variations. So if you open the alignment file, what you see will be like a list. Okay, you have the ends actually supposed to be the reference sequence, but if you don't provide reference sequence when you're trying to see it, it won't show you. You can show ends. All the green things, those are reads. So, and if it's not green, I say this blue one here, S, that means you have different alleles. So if you move down here, it has G and C and C and G, C, C, G. So, okay, for this locus has C and G, has 2 alleles. And the K here has T, T, G, G, okay? So you can see those. So those are possible polymorphisms, but how the challenge is how we can separate them from the noise. Okay, so things to consider here. We want the read that have very good mapping qualities. Most of the cases we use 30. If you want to be more conservative, use 40. Yeah. So the mapping quality is related to how many mismatches in alignment and how unique the mapping is. And so how confident the variation record, you're gonna have a variation quality score. So instead of mapping quality score, you're gonna have another one in the VCF file. You're gonna see a variation quality score. So it's based on how many reads support the calling. If I have more reads, have 100 reads, that'd be perfect. And what is the base quality there? So it's, remember base quality is from FASTQ. So what is the base quality there? Let's say I have one allele support that show up, but the base quality is poor in the FASTQ. So that might give us some clue, and the variation quality of the score will be low. Okay, so for calling variations, we're gonna be using a tool called Genome Analysis Toolkit, called short GATK. It is a tool, it's a package developed by the Broad Institute. It's kind of becomes a standard for calling SNPs and the indels in the DNA and RNA data. So GATK is very powerful. It's not only calling SNPs, it has other tools for like quality control or SAM or band file quality processing. So, but keep in mind, GATK was designed to maximize the sensitivity in order to minimize the false negatives. So that means GATK gonna be very generous giving you SNPs. So you need to run a lot of filtering to remove, to own or to pick the high quality ones. Okay. Is any questions so far? Oh man, I should be looking at this. Okay. Okay, so those are the introduction about the NGS, and we're gonna do some, after this, we're gonna do some hands-on session. So, but before we do that, let's take a 10 minutes break. Let's see, it's 11:23. Let's come back at 11:30. So if you want to go ahead and yes, so we're gonna do hands-on session. So if you don't have ADA account, please go ahead and apply. I hope you have received the email from yesterday and you already have the ADA account. So we're gonna use ADA, and we're gonna use some instruction I prepared and from the GitHub. So okay, let's take a few minutes break and we'll come back at 11:30. If you have any question, just send in the chat. Okay. So next we're gonna do a practicing how to calling variations from the sequencing data using ADA. It's part of one of the HPLC system. So first we need to log in to ADA. So if you have a terminal, you can use SH and your terminal EID at ada.tam.edu, you can log in, or you can use mobile Xterm. I think because Michael introduced all those tools in the last class, so I won't repeat it here. And you're gonna have all the instructions in this GitHub page. You can click and go to the page. So instead, another way to run those things is you can go to portal.aprc.tam.edu. It's gonna be on the web page. So still you need to log in to ADA and do the same thing. Let's try. So I'm gonna using the pro list here because I have a terminal, so I'm gonna using SSH. So everyone now just if you want to follow the practice, just go ahead and log into ADA. I'm gonna log into ADA too. Okay. So SSH, make this bigger. Okay. SSH. Yeah. Ooh. Yeah, to do, I need to answer on my phone. Okay. Okay, so I logged into ADA and give me login 4. So that is one of the nodes, one of the login nodes for ADA. So if you're using mobile Xterm, go ahead log in. And if you want to use a portal.hplc.tam.edu, here's how I'm gonna click. You're gonna show me the portal. Okay, I'm gonna use the ADA on-demand portal. Click on that. So it asks me to log in. I think it's probably the same dual thing. Okay, send me a push. Hello. Okay, I'm in. So we're gonna use if we want to use this one, let me see. I think it's a VNC, Interactive Apps, VNC. Do we need a VNC? Yeah, so number of hours, everything. We don't need a GPU, any GPU, any, let's say launch. Okay, when you click launch, it's gonna give you a screen like this, say your job is in the queue, and it should be pretty fast. Okay, so it's running, and launch. I'm gonna click on this blue tab here. Okay. So you got the X term. And it's similar. So I'm logging to now, but you see, if you pay attention to the node name, it's not logging something. It's NXT. It's actually one of the working nodes. So because in this portal, you're submitting a job and say how long how CPU you need, and you can assign you to a node, to a real working node. And when I use SH login to Ada, first I was assigned to the logging node. So that's the place you can prepare things but not running real jobs. Okay. Let me see if we can increase the font on this. This looks really small. No way. Okay. Anyway, so there's two ways. You can use a terminal if you have a terminal on the machine, or you can use portal.ada, and you're gonna end up with a terminal as well. Okay. So let's go to the instructions. Click this link, github.com, and that's where are you? Okay. GitHub.com. Yeah, I have too many things opened. Okay, so if you open the link, the GitHub link, you're gonna see a page here. It has all the instructions on how we run genotyping sequencing. So let's start from the setup step. We're gonna make a directory. So for all those things, you can either do it on the login node or do it on the, okay. So yeah. Yeah, the setup thing is fine. You can do it on the login node or on the working node if you're using the portal. But I have some steps like say download the tools and sample data. Those has to be done on the login node because the working node has no outside internet connection. Okay, let's do the setup step here. First, we make a directory and we're gonna build called genotyping under the Scratch folder. Okay, I think probably I already have one, so genotyping, yes. I'm gonna remove this guy to something else, genotyping.old. Okay. Command, we make a directory. So once we are done, we change the directory. We cd by say change directory, cd. Okay, now if say print current directory, you're gonna see this is my current directory. Yours might be different because we have different EIDs. So it's gonna be Scratch user, your EID, and genotyping. Okay. Okay, well, yeah, I prepared the tools and the data and everything. So for that to work, you have to run this on the logging node. So you have to use mobile xterm or SSH on a terminal. Okay, let's first download the tools. So there are two ways to download the first tools. You should just pick one of them. Let's pick a second one. looks shorter. Okay, if I will say use no check certificates, use the first one. Okay, the ls, you see the tools.tars here. Let's get the data. Okay, we need to use the no check certificate thing again. Really? Okay, so what you do, you just, let me try see this will work. Just add a no certificates. Yes. Okay, so see here, instead of using list, you need to add the no check certificate parameters after this wget. Don't forget the space. Then it'll be fine. You'll get everything we need. Okay, this might take a few minutes because many people are doing this. So if you have already downloaded the tools and the data.tar, can you please just put the Y on the message on the chat so I know how many people already finished. So far, I only see one person finish downloading the tools and the data. Let's just wait a little bit more. Okay, 2% finished. 3, they're counting. 4, 5. 5, 6, 7. Ooh, fast. It's 5, 6, 7, 8, 9, 10. Okay. Oh, that's right. Yes, you can also click on the yes on the participant page. Here, have a yes. Oh, 3. So when you finish downloading, when you try typing ls, you're supposed to see data.tar and tools.tar. So after this step, you don't need access to internet anymore. So you can run the following practice either on the login on the next node, on the working node. Okay, you have 13 people finished. If we get to 15, I'm gonna continue. Okay, if you have questions about downloading, please leave message in the chat so we can help you. If not, let's move forward. Okay, so we downloaded 2 files and they're in tar format, and tar is like a packaging format. So it's like container, put one thing in container. Okay, we need to untar those and put them in the right place. Okay, so you can just copy all those commands, but I'm gonna explain a little bit to you. So first one is tar xvf, it means x means extract file from this tar. So you're gonna run this one and you're gonna generate a folder called tools, and we're gonna move tools. I'm gonna generate 2 folders, tools and proscripts. I'm gonna move all those 2 into the home directory. Okay, let's do that and copy those 2. Okay, you see it's generate proscript and generate tools. And if I hit enter again, I already have them. So it gave me those errors. But if you don't have those tools or proscript, you're gonna just have nothing else. Yes, perfect. Okay, so next we're gonna get the data. We're gonna tar xvf and data.tar. Okay. Oh yes, so if you want to clear screen, get so many information on the screen, can use CLEAR, clear. We're gonna give you a clear screen. And ls. Okay, so now you see I have the FASTQ file as contains the, actually I can take a look, FASTQ slash, you can see it has the sequence data for samples. And I have sample 1, r1.fastq.gz, sample 1, r2.fastq.gz. So each sample has 2 files. One file for read 1, one file for read 2. You can tell from the R1 and R2. So we have 10 samples here, and we got 20 files. Okay, so we have all the files in the FASTQ folder. And there's another one called chromosome 6 underscore reference.fa. Remember we talked about the FASTA file? So the FASTA file, sometimes you can see file name.fa. FA is in short for FASTA. FQ is in short for FASTA-Q. So when you see .fa, it means it's a FASTA file. So we can actually check this file by using command called more and chromosome 6 reference.fa and hit enter. Okay, so as we introduced before, always the file, the sequence name always start with the greater than sign, and follows the sequence name. In this case, it'll be a console name. Okay, so there's a question on the chat say why the command cannot be copied and pasted to a portal. It's actually, you can paste, but it's just 2 more steps. Oops, this. Let me find the portal. Okay, so let's say I have something here. I want to copy this one. Copy. You have to go to, instead of Control C, Control V, all those things will not work. You have to pick something from the from this bar, from this here. Can you see my mouse moving here? I think this is probably the one. Yes, clipboard. Is that clipboard or something? C, Control V. Oh no, not clipboard. Something else. Let me see. We have a way to paste. It's just it's not very convenient. What's this? Oh, is that clipboard? Control Alt. Escape. What's this? I remember there was a way to paste into this, but it's just couldn't find it now. It was supposed to be a click. So let me ask Michael here. Michael, if you know how to paste into the mobile XM, can you put it in the chat? Hey. Okay, so paste in the clipboard first. Paste here. And what? Clipboard. Okay, I will let. Now you can paste in the terminal. What do you mean paste in the terminal? Okay, I'll let Michael take care of this. So, because I'm not using Portal very often, not really how. I remember there is a way to paste, copy paste, but it's not very convenient. So, if you have a question about the copy pasting mobile XM, you can direct message Michael Dickens Charles Michael. Okay, so let's go back to the FASTA file we're looking at. We have the list line start with the great than sign and following the name. So this case name is just 6, it's quantum 6, and has a lot of information to tell you what this is. It says DNA, repeat masked, primary assembly, a lot of things like that. Okay, then following that line, you're gonna be the real sequence, ATGC. Sometimes you see ANDs. So ANDs can be repeat masked because it could be repetitive sequence and it be replaced with AND. Every real sequence, and then keep going. So it can be a really long file. It's a lot of repeat masked sequence. So to quit list screen, you can push Q and go out. So when using more and use the space, you can go down, and you want to quit, just push Q. Okay. To clear screen and use CLER. Okay. Okay. So we have the FASTA-Q. We have the chromosome reference.fa, the FASTA file. So next, we're gonna prepare those things, right? So prepare reference file, prepare reference genome. For doing that, we're gonna need to have the tool called bowtie tool, because we're gonna be using bowtie to run this alignment, and then we need to prepare reference that is suitable for bowtie tool. And we also need to produce some dictionary file and index file. So we're gonna be using the pcard over here and the SEM tools. Okay, so we need 3 tools for this step: pcard, SEM tools, and bowtie tool. So we're gonna load those tools into the working space. Yeah, we are working now. So by default, when you log in or when you are on the working node, you don't have those tools. Let's say if I type in bowtie 2, I suppose you will see this, say command not found. So, okay, that's because we haven't loaded the tool yet. So let's load all those tools. Module load, I'll copy those three command here and paste. I'm gonna load pcard first. Load SEM tools. Okay, load bowtie 2. Yes. Okay, that's clear screen. So we already loaded all the tools. If we type in bowtie 2, it's supposed to see the usage information, not the arrow say command not found. And let's try it. Bowtie 2, okay, that's good. It's print a lot on the screen. Yeah, you have to use the mouse to scroll back. Okay, another way is, let's just a pipe in the terminal. So you're gonna be say bowtie 2 and more. Okay. Okay, now you start from the first part of the message. It's telling you, say, no index query or output file specified, because we just type in bowtie 2. And it's giving you usage information here. You need the -x, maybe a bowtie 2 index, -1, read 1, -2, read 2, or you have single n, -u. Then the output, all those things. Okay, now use Q to quit. Okay, so the following commands is used for produce. First, the bowtie 2 index. You see the bowtie 2 build here. This is gonna be the command to build the index. And we're gonna build some dictionary file. And we're gonna do some using the pcard. The pcard is Java's program, so that's how we run a Java program. And we're gonna be using the SEM tools to introduce the index file, to produce the index file too. So it's a 3 command in together. Let's copy all those from the reference equal something to the end. Okay, let's paste here. Because those files are really small, it's for testing. So, and you can run it on the login node. It's okay. But when you have a really big file, like big reference genome, it's gonna take hours to build the index. You probably will need to submit the job to a node, to the working node. Okay, the building index gonna take a few minutes. It's done. Wow, pretty fast. Okay, good. Now we have, you see the chromosome reference.fa. Now we have chromosome reference.fa.1.bt2. So those all end with BT2 are the bowtie 2 indexes, index files. And we also generate chromosome 6 reference.fa.dict dictionary and FA index file. Okay, so we have done preparing reference genome. So let's say you got a new reference genome. It's not called chromosome6_reference.fa. To reuse those scripts is either just replace this part with your real reference file name, and all the others will be the same. So except the first reference equals something. Okay. Remember we talked about a pipeline before. So when we have the data, we prepare the reference genome. Then we're gonna working on the sequencing data, right? So we're gonna be doing is we're gonna run a quality control step. We're gonna remove the low quality bases. We're gonna trim adapters, and we're gonna using a sliding window approach to trim low quality. Okay. So here's a command for one sample. Okay, for doing that, you need to load this tool. We're gonna be using module load to load this trim-o-matic tool. So let's load this tool first. Okay, while it's loading, let's take a look at the command here. So we're gonna, if you want to run QC for one sample, it's gonna be like a list, Java and jar. This is the path to the trim-o-matic jar file. Then say it's a parent mode, and how many CPU, how many threads to use, and then the path to the FastQ files. And it can be FastQ, or FastQ can be zipped FastQ or FastQ.zz. So it's fine. You don't have to unzip it. Trim-o-matic will take care of that. So this is the path to the read one, then follow the path to the read two for this sample one, right? Then you have to say what kind of output you want. So I want the output first is the forward read, it's actually read one. And the forward read unpaired. Then the second read, I say reverse read, and an unpaired reverse read. So then the parameters for how to trim reads based on the, we're gonna remove 3 bits at the end, 3 bits on both ends. You're using the sliding window, it's the same parameters we have seen before, and the minimum length. Okay, so this is how you do sample one samples. But we have 10 samples. So you can manually do it, but it require you to copy paste this command and replace sample one to replace to sample two, sample one to sample three, so all those things. So instead, we can also use a share command just to share loop to do all this command. And it's pretty easy, so I have everything here. So you don't have to run this sample one, and everything will be taken care of for the second part. So let's copy everything here from the for loop to the done, and copy everything and paste. So basically, you're gonna do trim-o-matic for each sample in the FastQ folder one by one. Because the file, okay, again, the files are really small. This is for testing, and I can run it on the login node. The files are really small. So if you have a real sequencing data, the files can be 10 GB or something. You don't want to do this on this login node because your job is gonna get killed in the middle because it's taking too long to finish. So it's gonna be submission job. I'm gonna introduce you how to submit the job later. So right now we just get familiar with the steps we're gonna be doing. Okay. Okay. So we already loaded the trim-o-matic with a module load command. So good. Now we just copy paste all those things here to the command to the terminal and paste everything. What's going on? Sample 7, SQ.gz. Okay. What's up here? What's going on? Oh, that's a lot. It's saying cannot find some files. That's the Q, sample 7. Okay, I see why. So I asked the command to output into a new folder called FastQ under /QC. So because the folder is not there, so and the Trigonometric won't create a folder for me. So before we can run the command, let's make a folder. So it's how we do it. Make dir. Let me clear the screen first. Make directory fastq/qc. Okay. And hit enter. If there's no complaint, that's good. Okay, now we can rerun this QC for all samples and copy paste everything here. See, okay, it's working. I should upgrade this part then, update this part. Actually, I can update it now. Okay. So I added make directory command in the documents. So if you refresh, you should see it. So make directory first, then run this whole part. Okay, because we are doing this quality control one by one, so it's gonna take some time. And hopes to be done very fast. Okay, so all done. So we can take a look. It will be in the FastQ folder and QC folder. Take a look. Okay, if you do this command ls fastq/qc/, you can see the output of this command. You have sample1 underscore f.fq.gz. Okay, sample1 underscore fs. It means the unpaired is single. So I just use fs. Whatever you like, you can choose any name you like. So sample1 r.fq.gz. So yes, you see we have all output. Means we are done with the quality control step. So next, we just need to run the alignment. Okay, the BOTA2 alignment is actually quite easy. And let's try. I think we already loaded BOTA2. If you type BOTA2, you see the help message usage, we already loaded. So in this case, I think we already loaded. So how to run BOTA2? This is an example for running one sample. So BOTA2-X, you have the chromosome, the index, and you have the -1 means read 1. Remember, we have the f.fq.read, the forward, and -2, read 2. So normally, I just ignore those sample underscore fs, those unpaired read 1 or unpaired read 2, I just ignore them. So if they are very small percentage, normally like 1%, 0.1%. So many times they are very small. I don't bother to put them in the alignment. I mean, you probably have 5 million reads and you lost 200, you don't really care. And you give a read group ID there and choose the very sensitive, local, or whatever parameter the field you want to use. And I think because the reads are still short here for this case, we can use local. Local is like a more conservative way to do this alignment. And how many parameters, how many CPUs? Say I use 10 CPU and -s sample1.sam. So this is output. Okay, you see here, the alignment taking a lot of resources. So we will not be running this on the login nodes. and we're gonna run it with when we go to the later B-sub part. But you see how you can run it with this bolt2 command. If you are on the portal, you can run this command because it's not on the login node, it's actually on the working node. It's fine. So I'm demonstrating this on the login node, so I'm not gonna run this. Okay, so after we finish the alignment, let's say you have done alignment for all the samples. Yeah, we remember we have several steps of processing. So because we ask bolt2 to save the files in SAM format, see here -s sample1.sam. So we need to do SAM, convert SAM to BAM. We're gonna be using SAM tools. This is a command we can use. SAM tools view, and we're gonna be sorting the SAM file based on coordinates. And we're gonna add a read group name if necessary. So in this case, we don't really have to do this, add a read group name thing. But this is how you can do it if you want. So the step 4 will be the duplicates. And in this case, we're gonna, so this is the data from the whole genome sequencing. So we are not supposed to see a lot of duplicates. The PCR duplicate actually should be removed. So we're gonna be using this tool called Picot, and using this one of these tools within Picot called mark duplicate.jar. It's not only mark it, it's also you have the option say remove duplicates. It says true. Then you're gonna remove the duplicates. Okay, when we have done this, step 4, step 5 will be gonna run some local realignment. Remember, it's because of the gap in the alignment can cause some false SNPs. So we want to run a local realignment. We're gonna be using GATK, and we need to have the GATK jar somewhere, and which will be, you need to load the GATK before you can use it. So since we don't have data now, so we're not gonna run it. I'm not gonna show how to run. We have the parameters here and the command line here. Here on the step 5. Okay, so it's actually 2-step processing for this local realignment. You're gonna figure out the place that need to be realigned. So it's the first step. Then actually doing the second realignment for the second step. Okay, so once we have those done, we have the BAM sorted, add RG read group, and remove duplicates, and local realignment. Okay, it's like already 5 steps. So the last step will be mapping quality filtering. You can use SAM tool to do it, it's quite easy. You just need to supply the mapping quality you want to use. So you can use 20, 30, 40, depends on, most of the cases I use 20 because depends how good the reference genomes is. Okay. So finally, we finished the processing the BAM files. What can we do now? We can call variations. So in GATK, there are 2 variation caller. One is called Unified Genotyper, and another one is called Haplotyper caller. So the Unified Genotyper is old. I think it's not being recommended anymore, but it's not like it cannot be used. It's just very generous. I'm saying it's like GATK in general is very generous, giving you SNPs or indels or those things. So Unified Genotyper is very generous. So when you use this one, you're gonna receive a lot of variations, SNPs or indels. So you think about filtering. So Haplotyper caller is a latest tool, and basically it's running a local Novo Assembly. So this way the SNPs and indels can be more accurate. And so this is a recommended tool, evaluation calling tool from GATK, and the parameters for those tools are quite similar. You see, if you compare them, except the name, the tools are different here. The parameters -R, -I, all those things are very similar. See, so okay, so here is a general process you can run from the FastQ file to variation file. Okay, so this is, I have so many steps. and you do not want to run manually one by one, right? So I have some tools that can help you run this faster. So that's why when I provide you a download, there's a tools folder and the Perl script folder. So in the Perl script folder, and in the Perl script folder, there are several Perl scripts gonna help you run the Perl process. So it has align.perl, gonna be run alignment. Generate B sub, process, haplotype color, process v2. So all those things, unified genotype.pl. So how do we use those tools and how fast can that help us? Let's take a look. So first, we need to generate a list that generates a text file that lists all the samples. So I have a command here. You can, I have fastq, yeah, fastq.txt. Okay, whatever. Let's copy this command. Okay, so basically this command gonna generate a file called fastq.list.txt. We can take a look by saying fastq.list.txt. Okay, so it gives us sample 10. This is the read 1, sample 10, read 2, sample 1, read 1, sample 1, read 2. Okay, so it's a text file with the list. First column is the sample. Second column is the read file, the path to read file. So you don't have to use a command to generate this file. If you have a list, you can either make this file in Excel or something, right? Just make them separated by the space or tab. Okay, once we have this file, I have a script called Perl script generate bsub.perl. So this is gonna help you generate all the bsub file for all the steps, for QC and alignment, processing, and finally calling variations. And you're gonna generate a bsub file. So what is a bsub file? Bsub file is a file that can use to tell the computer what you want to do, okay? Say for the QC step, I'm gonna tell the computer I want to do QC. So I have to provide how many CPUs I want to use, how many hours I want my job to run, and where are the files, all those things. So, and this script is gonna generate 4 bsub files. One for QC, for alignment, for the whole processing, then for the variation calling step. Okay, so let's run this script here. Let's copy paste this one and in the terminal, see what you got. Let's clean the screen. Paste here. Okay, you got the user message. Say, how do you use it? First, this one you need to provide parameters fastq list. We already generated one, fastq.list.txt. So that is the one we need. And okay, so the number of jobs, leave it as it is, it will be fine. Job name, give it a name. And you will need to provide the align Perl. Remember, it's in your home directory Perl script, align.perl, right? So, okay, that's what we need. We need those align Perl and calling Perl script. It's gonna be, you can use unified genotyper or haplotype caller. It's both in the folder. And reference index, reference file in FASTA format, and reference index. In this case, in our case, because we made it the same, so it's the same file name. Okay, let's actually generate list, generate that bsub file. So we need a fastq list, and its name is called fastq list.txt. Fastq. .list.txt. Okay, so I need to make attention to fastq.list.txt, not underscore. Okay, say number of jobs, 10, job name, whatever you like. I'll say my job. Okay, align pro. Okay, this part is tricky. You can use the tilde sign and the pro script, then align.pro. You see here it has my net ID here. You can replace my ID with your EID. This is my EID. So to use it, you have to replace with your EID. Or you can just use the tilde sign here. The tilde pro script align.pro. Okay. I'm gonna need the call pro. It's in the tilde pro script. Let's say we are using unified genotype color.pro. And the reference genome is in the current folder called chromosome 6, right? Reference.fa. And the reference index, and likely we made it the same. So we just provide the same. You see here, here's a way to run this script and help you generate all the necessary bitsub files for you to run the whole processing, to run the whole variation calling. Okay, so if I hit enter, you're gonna generate a lot of files. Okay, and clear screen. Let's see what we got. Let's see all the bsubs. Let's say ls star.bsub. So it's generate 0 is a QC step.bsub, 1 is alignment step.bsub, 2 processing means process all the alignment files, and 3 calling variations. Okay, so we can take a look, see what's inside each file. Let's say more 0.qc.bsub. If you run this command, more is a way you can show the content on the screen. 0.qc.bsub. Okay, you're gonna see a lot of things. Those start with the hashtag, basically like comments, but the hashtag bsub thing is a parameters for the job scheduler. Here you're saying -w is how many times you want to run. Its format is hours, and hours first, and colon of minutes. So it is 70 hours, 5 minutes. It's just arbitrary. So this one you don't have to change. And the number of tasks in job, I'm going to be using, basically saying how many CPUs you want to use. I'm using 4 CPUs. And here is saying how you want your 4 CPU to be choose. So you can be, let's say you want 4 CPUs, but it can be 1 CPU per computer per node, right? So you get 4 nodes, and on each node have 1 computer. But for our case, the program we'll be using, they cannot communicate between nodes. So you have to have all those 4 CPUs in 1 node. So let's say p tile equal 4. If you have the p tile equal 1 here, you'll get 4 nodes and 1 computer and 1 CPU per node. So that's not what we want for this case. We want them all in 1 node. And the memories here, 5,000 MB, but 5 GB, and 5 GB times 4 will be 20 GB. So you're asking for 20 GB memories per node. And we have 10 samples. So it's gonna be run each sample in 1 job. And it'll be my job QC and 1 to 10. Okay. Other things like you have to load the tools you need, module load, all those things. And then you have to say, if it's job index 1, we do this. Job index 2, we do this. Job index 3, we do this. Okay. So basically, it's the same command. Here you see Java jar, trimmatic.jar, and parent mode, all those things. But instead of running them one by one or in the node manually, we put them into a bsub file and submit to the working node. Okay, so it's the same for the alignment, and we can take a look too. First, we got parameters here. How many CPUs? In this case, I want to run 10 CPUs, and the memories per CPU will be 2 GB. So in total, still 20 GB. And only ask for 2 jobs. Okay. Okay, so you see we have those bsub files ready. So how do we do it? So one way is we can submit B sub and what's that? The arrow sign, the less than sign, then 0.qc. So you submit the first step first. When the job is finished, then you submit the second step, 1.align.b sub. So in this case, but you have to keep coming back and check see if the jobs are finished or not. So it's or you can submit all of them in once. And the first job finished, the second job gonna start. Second job finished, the third job gonna start. Third job finished, the fourth one gonna start. But that's just assume there's no errors in the previous steps. So many cases, there might be errors there. I mean, the error doesn't mean something wrong with your file or something. It's just, it could be you are requesting, let's say you are requesting 70 hours, but the job actually took longer time to finish, so it's gonna be an error. Or you're requesting 20 GB memories, but the job actually used 30 GB memories, and your job gonna get killed, and you got an error. So sometimes you have to check the output, the log files to make sure everything's fine. If you see anything, it's not good, and you have to modify the parameters according to the error message you got. So in this case, we can run them one by one. And when one finish, and go back submit the second. So let's try submit the 0.qc b sub step first. The b sub and smaller than sign 0.qc b sub. Okay, you hit enter. You're gonna see. Oops. Yeah, you're gonna have some message. But eventually, you're gonna see the job is submitted to default queue as n regular, and as used to charge, that's based on how many SU requested. But it doesn't mean you're gonna be charged for this amount. If your job finished earlier, you're gonna be charged less. Okay, so we submit the jobs. So how can we check the status of the jobs? What's that, B jobs, maybe? Yes. Okay, if you're typing B jobs, okay, you're gonna see the information about the job. So here I have list. The first one is as a regular VNC. It's the one from the portal. I started the job over there. So it's gonna show up here too. And then have my job QC. I know those are the recent jobs I submitted because they start with my job underscore QC, and we request 10 jobs, so it can be 1, 2, 3, 4, 5, 6, 7, 9, 10. Okay, so you can come back and check this one time to time. Oh, say it's finished because the files are really small. I assume they finished pretty well, pretty fast. Okay, so you don't see any of those my job QC, that means it's finished. But before we want to know it's finished or not, we're gonna check one of those. You see, so each job actually produced two files. One is .err, one is .out. So let's check the .err first. Just any .err. And you're gonna see some of the logs, the messages, how the jobs were executed. So you have the following have been reloaded and everything. And to execute, come on, no. So, do I see anything wrong here? Let's check the out again. Okay, it looks like okay to me because we already run this command on the terminal, so we have everything anyway. For me, because it's generally so many log files, so many .err, so many .out, I just make a folder and move everything into the logs file. So then it looks cleaner. Okay, so we finished the first step. Sub. Now we need to submit the second step, right? B sub 1.align.b sub. This is how you submit the second step. We hit enter. I'm gonna bring you to the next step. Okay, you see a lot of message. Do I get to submit? Okay, It's the same method here. This job is submitted to default queue. And if we say B jobs, we see we only ask for 2, but we could ask for 10. So we only ask for 2. Anyway, they are running. And the alignment file gonna be saved in the alignment folder. So let's take a look at the alignment folder, ls space alignment. We got nothing yet. So, and we try B jobs, they are still running. So just no files being generated, but the job it's already there. Okay, so let's say when this job finished, we're gonna submit the third one, right? It's gonna be sub-2 processing step. And once it's processing step, we're gonna submit the variation calling step. Okay, so that's how it works with B-sub. And I don't think we have time to waiting for all those steps to be finished, but I encourage you to try the following steps with B-sub and see if you get the final output in VCF format. Okay. That's all for the hands-on practicing step, and all the instructions and script tools you can find on this GitHub page, and it can be easily modified for your future use. You just need to modify to change the reference name, to change the job name, all those things. So you can either modify to adapt to your future project. Hope it can be useful to you if you are still keep using, if you want to keep using the ADA system. And I think that's all for today. And if you have any questions, I'm gonna happy to stay for 10, 15 more minutes. Okay, I have a question from the chat. Say, how do you move a group of files into a new folder? So you probably see me. I move all the .err.out to a new folder called logs. So I just move say mysql.star. So it's a wild character here .star, and I'm gonna take everything as match this pattern. Star means whatever you got after this. To the log file, I do this. I hit enter, gonna move everything start with this, end with anything to this folder. So there are many ways to do that. Let's say, yeah, basically you have to use the regular expression to select multiple files. Yeah, I'll give you an example here. Let's say, how do I select all the thing with a chromosome 6? So I can say ls chromosome 6 star. You're gonna list everything, all the files start with chromosome 6. And you can see those, right? If I want to choose everything ended with BT2, I can say ls star.bt2. I'm gonna select everything ended with BT2 with this command. So inside list, you can move it, right? I can move those things to some place, or I can copy it to some place. So it's the same idea. I see there's one asking, will the recording of today's webinar be posted anywhere? I think we do have recording posted to the YouTube channel, HPRC YouTube channel. You can probably go search there. But it's gonna take a few days. I remember it's gonna take a few days. Okay, everyone, enjoying the weekend. Stay home, stay safe. We'll see you next class.